In this video, we'll talk about proteins. Now, when we're talking about proteins are throughout the whole body and they do a number of things. And we could break the world of proteins down into two categories when we're looking at structure. So in terms of structure, some proteins we can consider fibrous, and the name is very descriptive. It gives it this long rope-like strand appearance, and it's mostly non-polar amino acids that are part of this. Now, these fibrous proteins, what do? They give us strength and durability to structures. The other structural way we can look at proteins is those that are globular. And once again, the name gives you some idea. It gives you a sphere or globe-like. And this is mainly made of polar amino acids. And these are things that we see function as enzymes and hormones or cellular messengers, I should say. Now, there's a second way. So we just looked at the structural way, but if you think functionally, we could also consider proteins in two categories. One category would be basically the proteins that do things, like enzymes. So they're sort of these action proteins, and the other category could be structural, that they're the ones that form structures. So you could either look at proteins based on the structure or the function and divide them that way. But here we're talking structure, and we'll consider that there's fibrous and globular proteins. Now, when we look at proteins, they have a hierarchy of shape and organization. That, of course, all proteins are made from a chain of amino acids, and we can see that happening here. Here they're representing a chain of amino acids, and we can see different amino acids like glycine, serine, cysteine, and they're joined together by a peptide bond in order to make this long string. Now, the first level of organization for a protein is a primary structure, and the primary structure is just the amino acid sequence of the polypeptide chain. Now, this is not the final stage of being a protein, because in the world of proteins, it's all about your shape. Proteins have very complex shapes. They fold back on each other based on the amino acid sequence, etc. So the primary structure is just merely the structure we have when we hook together a whole bunch of amino acids. Secondary structure. This is where we start to get some of that folding or the bending or the twisting or the interaction of different parts of that long string of amino acids. So when we're talking about secondary structure, one or more segments of the primary structure get folded into specific shapes. And this is very specific because, as I said, if you want to be a functional protein, you need the exact shape for things to happen. And we have two sort of ways that the parts of this primary structure can fold or interact with each other. And one we'll call the alpha helix part. So in the alpha helix, we begin off with that long chain of amino acids, and certain amino acids would like to interact with other amino acids along that chain and form bonds. And if that happens, and let's zoom in, because here's a long string of amino acids, and this amino acid would just love to interact with over here. So what happens, or up over here I should say, so what happens is that we have a folding to allow that to occur. Now sometimes this fold gives us a spiral-like look to it, and we call this section that takes on a spiral-like fold an alpha helix. Sometimes it folds in a way that we call the beta pleated. And the beta pleated is, imagine taking a sheet of paper and folding it over and over and over. Those folds where different parts of the original paper come in contact with each other would be very much like a beta pleated. So when we're looking at a protein, it actually takes on quite advanced structure. And it's not just an either or. that here we have the primary structure, and in some cases it's done the sheet folding, the beta sheet folding, and here it's done coiling. So often in a finished protein, we'll have areas that are the beta sheets and also the coiled areas also. All right, that's secondary structure. Let's move to the third level, which we call tertiary structure. And this is the three-dimensional shape of the polypeptide. Now, even though we've undergone, and let's zoom in here. So here we can see that we have really advanced folding. So remember, the primary structure was just that long string of amino acids. And then what happened is we start to get beta pleat sheets occurring. We had coils occurring. And then this whole thing is now going to start to fold in on itself even more, kind of like origami. And we're working our way towards the tertiary structure. So you can see here in this protein that there are regions that are beta sheets throughout here. There are regions that are the alpha helix. And after that, you can see that the protein folded back on itself even more. And in this case, it's made a globular protein. Over here, here's a fibrous protein. You can see that they really contain a lot of these alpha helixes. So we have the primary, the secondary, tertiary. And let's talk about the next one, which is quaternary. Now, not every protein goes to the fourth stage, which is the quaternary. This happens when I get a linking together of more than one polypeptide chain in a very specific arrangement. Now, this is often critical for the function of the protein that has to join together. So when I'm talking quaternary, it's the assembly of two or more polypeptide chains into a functional protein. And as I said, do note that some proteins do not, and many proteins do not have a quaternary structure. They don't need to go to this step that the tertiary structure is its final form. Okay, and here we can just sort of see the overall, everything sort of summarized here, is that, remember, the primary structure is just the chain of amino acids that are linked together by peptide bonds. And then we have secondary structure, which we start to get the interaction between various amino acids because of their chemistry and their side chains. They want to fold and interact with different ones along this chain. So maybe that one and this one want to interact. So the only way they can do that is by folding or twisting so they can actually come together. And that is the secondary structure. And remember, secondary structure has alpha helix or can have a beta pleated sheet. And in a given protein, you're probably going to have some regions that are beta sheets and alpha helixes mixed in. The tertiary structure, once again, is the further folding beyond the alpha and beta folding. And then the quaternary, not every protein goes to the quaternary. It's where two polypeptides join together for their final function. Okay, now proteins, because they're all about their shape, it's very important that the shape does not get destroyed. And if a protein changes its shape where it's no longer functional, we can call that denatured. The protein is denatured. So that's destroying the protein shape. Now, what types of things will do this? Heat will do this. Changes in pH, exposure to chemicals like change in salt concentration, etc. And when you start to denature, what are you really doing? You're starting to break those bonds that were used to hold together the different regions of that polypeptide. So we're going to start losing our structure. So I disrupt hydrogen bonds and ionic interactions that are really the hallmark of holding together that very complex protein shape. Okay, we're going to leave it there when it comes to proteins, and next time we'll come back and talk about nucleotides and nucleic acids.